if you think about the microbiome in the chicken, there's thousands to potentially tens of thousands of species of bacteria in the gut at any given time. And it's difficult to take the totality of that into one profile and say, this is good, this is bad.
Welcome to the Poultry Podcast Show. Today I am here with Dr. Tim Johnson. Welcome.
Hi, thanks for having me. It's good to be here.
I'm really excited to chat with you about vaccines, the microbiome, some of the things that might change outcomes. But first I want to hear about kind of your history with poultry and where you got today. How did you start your work with chickens?
Yeah, my work with chickens goes back to North Dakota State University in about 2000 or so, that right around the turn of the century, I guess, which makes me sound old. But anyways, I was doing a PhD project on, I had no prior experience with birds from a disease standpoint. And my advisor, Lisa Nolan, who is now the dean at the University of Georgia, was working on colibacillosis in chickens and laying hens. And guy got wrapped into a project there that was studying the genetic traits that make E. coli successful in the bird. And that really started down a path that hasn't ended for the last 25 years of an interest in bacterial diseases of poultry, but then that also parlayed into the microbiome as well, right? And so we've tried to learn quite a bit over the years about just what a normal gut microbiome looks like in a chicken or a turkey, and how you can potentially modify that in a positive way. And then that all ties back to diseases like colibacillosis and others like necrotic enteritis, et cetera.
I can certainly say that is an amazing area to be working in. It's always relevant because it's a constant pain in the side of the poultry industry.
It's fun working with the persistent chronic problems, but then all of these cool, interesting things come up along the way too. There's never a dull moment, right?
Boehringer Ingelheim develops poultry vaccines of unparalleled quality, with a focus on prevention and control across a wide range of diseases. We deliver unique, sustainable solutions for broilers, layers, breeders, and turkeys. Our veterinary network brings unsurpassed knowledge and experience, helping producers make smart, innovative, science-driven decisions to improve bird health and performance. For more information, visit poultryhealthusa.com.
Can you tell me a little bit about the microbiome? I know it's hugely kind of been changing as our diagnostics and our ways of analyzing and sampling methods have changed, but can you kind of give us an update? Like what would you consider kind of the state of the art for sampling and determining what's normal?
It's been a long journey, I think, from if you think back to the early 2000s when the ability to sequence the 16S gene started, and that now let us, instead of just being able to look at what we can culture, we could look at everything that's out there, with a lot of it being things that we have no idea what they are, right? Which that was exciting in itself. It was kind of like walking on the far side of the moon and trying to explore and understand what those things were. But the technologies have really advanced over time. They've become more cost effective. And I think the biggest difference now is we have better databases to classify what's there or who's there. But we're also expanding into this field of the entire metagenome. So not just who's there, but the genes that they contain. And then even the functional microbiome, which I think is the more recent advance. And that's the ability to not necessarily not care about who's there, but to be able to understand what they're doing in the context of who's there, if that makes sense. And so initially, I think we really focused a lot on just defining what is the baseline microbiome in a chicken look like. And now we're really kind of changing more towards, well, we know that there's a lot of variability in that baseline microbiome, but what is it? What are they doing? What role are they playing in the gut?
Yeah, so it seems pretty clear that the variability is huge in microbiome. It might be more specific to the specific location you take it from in terms of the poultry house, but also the region of the gut.
Yep.
So if someone is trying to understand or interpret, let's just say a research paper, or a salesperson brings you some data and you're trying to understand, like, how does a layperson know what to do with that kind of data? Like, what are some maybe best practices for interpretation of that information?
That's a really good question. And I think we struggle a lot with that, actually. And I think that's still kind of the missing piece, because a lot of people just want to know, is it good, bad, or other? And if it's bad, how do I make it good? Right.
Yeah.
I'll be brutally honest here. I don't think there's a way that we can say specifically this is good or bad, but what we can say is something doesn't look normal, right? So there's a lot of, if you think about the microbiome in the chicken, there's thousands to potentially tens of thousands of species of bacteria in the gut at any given time, and it's difficult to take the totality of that into one profile and say this is good, this is bad. So what we tend to do is we take the scientific literature that has been done and look for these markers, and usually it's a smaller subset of maybe a couple dozen markers that we tend to correlate with good or bad, and then we use those as the profile, which seems a little simplistic because we're kind of in some ways ignoring the rest of it. But I think the truth is we just don't fully understand the rest of it yet.
So what are some of the major influences then on the microbiome?
That's something that's been studied quite a bit. So there, basically everything has some kind of influence, right? But we can tier it in terms of what drives changes in the microbiome more than others. And so from what we've seen so far, the biggest drivers, and it's not something that is like groundbreaking or anything. It's things like, the biggest one in my mind is, especially with a young bird, is just the age of the bird. So as the bird ages, we see this rapid maturation of the microbiome in the gut, and it occurs primarily in a broiler between 0 and 21 to 28 days. By the time they get to 28 days, it really becomes what I would consider a mature microbiome. And so that's that. Those first 4 weeks of age, those changes dominate more than anything. But then when you're thinking about management, what we've seen is that even things like, so diet is a clear one, right? So changes in like large scale changes in the formulation of the diet, say a wheat based as opposed to a corn or soy based, is going to have a pretty dramatic effect. But if all things are equal in the diet, then really it comes down to things like season, time of year. Which doesn't seem like it would have a big influence, but interestingly enough, we did this study where we looked at one vertically integrated broiler operation, many different farms within that operation. Then we looked at the multiple barns at the same time growing, and then we also looked at multiple barns across seasons and flock cycles growing. And what was most interesting was that the time of year, and was the bigger driver as compared to like two barns that are geographically separated, so within the same system. And I think what's going on there was, it's not just necessarily time of year, but probably also the influence of the hatcheries and those other upstream parts of a vertically integrated system have a dramatic impact on what you see at the barn level. Because just intuitively you think, well, the barn environment is going to drive everything, but in fact, it seems as if it's upstream activities that drive the barn environment, if that makes sense.
Oh, yeah. Yeah, that's really interesting.
And then you get into the world of feed additives and vaccines, and those all have influences. But what we've learned is that once that baseline gut microbiota is established, it's really difficult to set it off on a totally different trajectory. So
yeah,
there's a baseline that's just going to be there no matter what, because of the environment and because of where the birds were hatched, and probably something to do with the parents as well. But these feed additives like probiotics, prebiotics, phytogenics, and even postbiotics all can have an influence, but it's minor compared to the influence of that baseline.
Honestly, it's really interesting because we think about the place they spend most of their time, but the influences prior to that are really, really important.
Really important, I think. Yes.
Yeah. So in terms of the makeup, we can always think about like what can and can't be influenced. There are some things we just can't change. There are some things we can. Mm-hmm. Is there a normal balance of like protozoa to bacteria to fungi in a healthy microbiome, or is a healthy microbiome just what works in a bird, an individual bird?
I think there's two things at play there. So there's the balance of the different bacterial species plus the non-bacterial, like you're talking about, protozoa, fungi. So there's definitely an expected balance with that. And so, for example, when I come into a 7-day-old chick, I would expect to find a lot of proteobacteria, very few Clostridia, very few strict anaerobes, and hopefully a lot of lactobacillus in the gut, because that early on, that's a healthy composition, right? And you will find fungi and you will find viruses. There's definitely an interplay between those three, but I think that aside from a fungal pathogen being present, I don't think that those other populations necessarily are directly at play with one another, right? With the exception of things like phage, which are clearly viruses that are going to have an impact on the bacterial populations. But then when you go into a slight 2 weeks from that point, so say 21 days of age, at least in the Zika, there's a big shift. So it starts being dominated by lactobacillus, and then it changes over those 2 weeks to being dominated by firmicutes primarily. And you see then, as kind of a third stage, you start to see Bacteroides specifically coming in, and we talk a lot about that ratio of firmicutes to Bacteroides as being, that magic number. Now, I don't know how much that applies to chickens as opposed to humans necessarily, but we know that we expect these things to come along. And actually, what I've done in the past is you basically set your baseline as to what you expect to show up when and where and in certain abundances, and then you can track that. So if you're looking at, does a feed additive have a positive effect, it's not necessarily do I completely shift the microbiome, but it's do I set it on the trajectory to maturation faster? That's what I'm looking for, is do I see those markers appear earlier and in higher abundance than I would see in a control, for example.
Yeah, that's a really interesting point, is that maybe it's not the total composition, but the speed, the trajectory.
And then the other thing at play is that we tend not to look at is just the overall load, right?
Oh, sure.
So with microbiome, we're always looking at relative abundance. So what percentage of lactobacillus do I have compared to my percentage of E. coli, for example? But we're not typically looking at just like what's the overall bacterial load in the gut. And I think that is a huge thing too. And actually, when you think about feed additives, I tend to think that they're trying to also, probably even more so, keep bacterial load at bay as opposed to necessarily targeting specific groups of bacteria.
Yeah.
So it's just another thing to consider for sure.
Yeah. Yeah, that is, enumeration has got to be quite the task. to get the total amount compared to percent abundance.
There's a really good old fashioned way. You just go in and count the cells, right? But
Yeah.
that's not a lot of fun.
Yeah. I'm just thinking about the number of samples you take, the location, and then a poor grad student sitting there plating everything. And
there are some ways with 16S where you can kind of semi-quantify how much is there to see if there's maybe more in one sample versus another, but it's not absolute, but it can give you an estimate at least. Yeah. So at least you can run a PCR that's a little easier than counting. Yeah.
So just kind of like thinking about who's there and in what amount, like what are the environmental factors or stressors that might change those outside of, just a normal environment? Like what things are known to change something about the bacterial load?
I think, one that we've been interested in recently is the idea that vaccines can have both a positive and a negative impact on the microbiome, depending on what they're doing. And so in general, what we've seen are there, as the poultry industry people well know, there's some vaccines that create an adverse response by the bird initially, right? So there's the classical one is hemorrhagic HEV vaccine in turkeys. And that is the way that they apply it in the industry right now. It's known to elicit a pretty strong response in the birds and even a dysbiosis in the gut potentially. So I think, that's the obvious example where you could have negative effects if you vaccinate and it actually, the bird's trying to build immunity, but it's also reacting to what the new antigen is encountering. And as a result, you can actually have indirect changes in the gut. That's one way. But the way we've been thinking about more recently is this idea that vaccines that are not eliciting a strong adverse response in the host actually work through the gut-brain axis, right? So if you're training your immune system, everything it's encountering is interfacing with the nervous system, right? And so you could be vaccinating with a respiratory vaccine and having an influence on the gut microbiome in some way. So, and typically, I guess classically, it's been thought of as a bad effect. But what we've been seeing is that some of the respiratory virus vaccines alone, and then plus if you're doing a combination of vaccines that might include Coccidiosis, vaccine as well as a respiratory vaccine, they actually seem to be over a window of time, they seem to be positively influencing probably the gut integrity. And also just as a result, I think that has this influence on building up a strong barrier in the distal gut that's helping to protect, but it's also at the same time it is influencing that microbiome, right? Because what you want in the case of Cocci and other pathogens that are in the gut is you want that really strong gut barrier. And the only way that occurs is if you have those good bacteria protecting against foreign, pathogen invasion, as well as the structure of the gut being strong. And so we've seen some positive influences with these vaccines that will, my example before about accelerating the gut microbiome maturation, some of these will actually do that. And then in one of the more recent projects we did, it was looking at a Cocci vaccine with and without a Newcastle vaccine. And what we found was that the whole goal was to come back and challenge, right? We were going to, they were looking at the vaccine and then come back with a challenge later. And so that was, obviously you want to see protection there. But what was interesting was we saw in the microbiome leading up to challenge was that there already were changes occurring in the vaccinated groups that seemed to be accelerating the appearance of those Bacteroides earlier. And So if you look at the overall, like back to my thing about, does it really shift the microbiome? It's a subtle shift, but it's a shift in a way that's moving it towards that mature state faster, I think.
So then what is the impact of maybe a stressor could even be not having food overnight? You get rid of the digestive tract contents pretty quickly in a fast growing broiler. Is that something that's stressful? Is that something that impacts makeup of the microbiome and how other things?
Yeah, I mean, there's a lot of things, I think, but that's a great example. Any type of feed withdrawal or any type of, even things like just how well are you maintaining the litter environment in the broiler farm? Right? I mean, that has a huge impact, right? Because what is on the litter is it's what they're basically pooping out, right? And when things fester in that litter environment, they change quickly, right? So things bloom here and there. If you're doing proper maintenance of the litter, you're going to have less of that occurring, I think. But again, anything like that can have an influence. Water can have an influence, particularly with turkeys, where they drink out of the drinkers, right? Biofilms are going to form in those drinkers. Even if you chlorinate the water, you're still going to get biofilms in those drinkers. And if you don't manage that, you can get a lot. There's been a lot of things we found in drinkers that I would not expect to find alive in drinkers. Bacteria that were just very surprising, and even viruses as well. So I think that's something to consider. But certainly, everything that you can think of that relates to management will have an influence. And then anytime we talked about kind of big, large scale feed changes, but even especially with things like probiotics, everyone tends to think of a probiotic as a good thing, right? But just remember that there's many different strains of probiotics that are being sold, and the vast majority of them are not derived from a chicken. They're coming from cheese, or they're good. They do good things, but they're not necessarily going to have a really strong influence on the gut of the bird, and sometimes even have negative influences, right? So that's another thing to consider.
So speaking from a purely microbiome standpoint, because there are many reasons that we vaccinate, on day of hatch before they get on feed. From a microbiome standpoint, is that a good practice to give vaccines at the hatchery before we really have a lot of exposure to environment? And then, we even give cocci vaccines, which are meant to cycle, and there hasn't been food through the intestine yet. So is that a good or a bad kind of microbiome outcomes?
I don't necessarily want to say good or bad, but I think for sure if you're considering that you want to establish that gut early, and you also want to train it at the same time, I mean, there's definitely a need for those early vaccinations, right? I mean, really.
Yeah.
But is it ideal in the sense that we're timing it right from a microbiome standpoint? Probably not necessarily. It might be better to, and some of these technologies they've got, and I'm thinking more on the turkey side, but these hatchery technologies they've got have the ability to do oral gavages, right at.
Oh yeah.
And so they're now starting to do things like give them a complex mixture of microbes right at the hatch, and then we'll vaccinate them at the same time. So I think that's getting a step closer to trying to time things appropriately, right?
Yeah.
But certainly when, traditionally if you're putting birds on a truck and you vaccinate them and then send them off, and you might give them some grow gel or something like that to keep them hydrated. But I think definitely it's something to consider, is that maybe that early, even, just being able to provide nutrients early on that would benefit the gut and the gut microbiome is something to consider.
Yeah. So with those probiotics or just exposure to other viruses or protozoans or whatever, like, are there some known influences on the microbiome? I mean, are there viruses that we know have a negative impact, or other? I mean, what are some of the things that, just kind of microbiology speaking, in the first few days are influencers?
Yeah. The biggest, I'll just generalize a little bit. So the biggest thing I think is, and depending on if you're talking about vaccines or just actual wild type viruses, right? But if you're talking,
Either, yeah.
if you're talking about the wild type viruses that may not cause necessarily mortality, but they suppress the birds in some way, right? Depending on how they're suppressing the birds, it will have an impact on the gut microbiome. So if it's an enteric virus and it has a weakening effect on the gut lining, that really opens the door. And this is a typical thing we see, is that you get an enteric virus that comes in, and what follows from a microbiome standpoint is what we call a proteobacteria bloom, where these proteobacteria, and that includes things like E. coli, Salmonella, Klebsiella, Enterobacter, they when the gut goes haywire for whatever reason, they have the ability to just blow up, essentially, because they're what we call facultative, right? So they have a variety of ways to use energy and to use or not use oxygen, and so they just have this ability to thrive in poor conditions. And I think that's a very common thing you'll see. But the other one would be, you get immunosuppression because of maybe a respiratory virus. And the immunosuppression itself will have an influence on that trajectory that the microbiome is going. And we've seen this with, coming back to that study with the broilers that I was talking about earlier. There was a farm that had a pretty significant E. coli problem during the study. And when we were sampling birds, we were sampling healthy birds and checking them to make sure there was no signs of disease or anything. But even in those healthy birds from the same farm system, they had a gut microbiome that it should have looked like a 21-day-old gut microbiome, but it looked like a 7-day-old gut microbiome.
Oh, wow.
So even though you didn't see disease, I think just that suppression with the pathogenic E. coli was having an influence across the entire flock, and it was just stopping the movement of that gut microbiome towards a mature state.
Yeah, that can be so difficult, because you might just have birds in the group that look like they're physically okay, but they're hiding that they've really got some issues.
And that's where I think there is a place in the future. I mean, I think there's been a lot of interest initially in microbiome research, and then in the poultry industry now it kind of turned towards skepticism, because it's difficult to tell somebody, what does this mean? Is it good?
Yeah.
Bad? Can you do anything about it? Right.
Yeah.
But I think now we're getting back to that point where, coming back to the idea of markers, I think there is a place in the future for a more customized, field level microbiome profile, where you can come in and take a very quick profile of a flock microbiome, and then try, with enough science, you can then give them recommendations on, well, here's what we know can change this in a positive direction, right? Give them some options, right?
So, so in terms of just, flock health and the way that we vaccinate, we've got different routes. I mean, some of them are, they go through feed or water, they're oral. Some are sub-Q or other methods on day of hatch. Is there a general difference between those two in terms of impact on microbiome outcomes, just based on route, not even talking about the type of vaccine?
Well, route certainly will have it. I mean, I would say in general, you're probably going to have less of an influence on the gut microbiome with a spray virus, right? Like a spray vaccine, and that's targeting the respiratory tract. That doesn't mean there will be no effect. It's just whatever site you're targeting is where you're more likely to have a microbiome. And don't forget, there's a microbiome in the trachea of the bird too, right?
Yeah,
So we tend not to even focus on that, but and in fact, it's dominated by a lot of lactobacilli, which is kind of surprising, right? Because you would expect things like, staph and strep, which are there, but actually there's some of the dominant bugs there are lactobacillus. And what we've seen in that sense is that you could vaccinate a spray vaccine that could have an adverse effect in the respiratory tract. And you see even more dramatic shifts there than you would with something going on in the gut. Like if you were to give a harsh vaccine response in the gut, there will be a shift there. But the respiratory it seems like it's almost an all or none. Like it either looks really good or it looks really bad. And there doesn't seem to be much of an in-between. And so I just think that's fascinating. We haven't really looked at it very much at all. So
Yeah, for all of the respiratory diseases that cause issues, we pay so much attention to the digestive tract.
And then coming back to your point about the type, I think the live attenuated seem to have more of a direct or short impact on the microbiome than necessarily like a killed or subunit vaccine.
Yeah, I guess that definitely makes sense because those live attenuated are trying to do their job.
And some of them are designed to colonize and then hopefully disappear, right? But
yeah,
by colonizing, they're influencing the microbiome for sure.
So for some of our diseases like coccidiosis, we have different methods for control. We've got oral vaccines, and then a producer might, cycle through later, different flock, different time. They might use like chemical or ionophore. I could imagine that's got some sort of different impact. Have you looked at or studied any of that?
So we haven't looked at them all combined together, but we've looked in bits and pieces over the years. I've looked at the influence of an ionophore as compared to maybe later on we look at a live attenuated vaccine. And all I can say is that the response, the effects on the microbiome appear to be distinct between different classes, drugs or vaccines that you would use to control. It's not most certainly not just a uniform effect on the microbiome. And recently, a lot of people have been talking about maybe the vaccine would be more effective if there was a proper microbiome or the right composition pre-established. And that's like, and it comes back to your point about early vaccination, right? But there's some evidence out there that if you think about, in humans, we always talked about the anaerotypes of the gut, meaning different people have different, healthy people from different places have different cohorts of microbes that live in the gut, but they're all considered healthy, right? We're finding some of the same things that if you take animals that have different anaerotypes or different cohorts of bacteria and you vaccinate them with the same vaccine, you actually get stronger responses with some anaerotypes as compared to others. So what it is that there probably is a way that we could balance the microbiome in the right way prior to vaccinating to get more of an effect with vaccines.
Yeah, that's so interesting to think about, but a complex research trial.
Extremely complex, and that's the problem with any of these feed additives vaccines is that you can study one or two or maybe three at a time, but it gets really challenging to uniformly study a whole bunch of them alone or in combination. And that's what I think we really need. But I don't think there's funding that could cover all of that, unfortunately.
Gosh, plus your comments about the time of year. I mean, you have to block it by season.
There's so many confounding variables that, yeah.
Gosh, it's unreal. Do you have a favorite discovery or something that you've had over the years regarding the microbiome that just made you want to continue to pursue this very difficult area of research?
One thing that was really interesting when we first started looking at the turkey and the chicken microbiome, and this was it was just about 10 or 12 years ago. And we found that there was a specific bacteria in there called, it's a candidate bacteria. It doesn't even have a species name, but it's called Candidatus arthromyces, which you think arthropods, but it's actually this It's the strangest bacteria. If we looked at it under a microscope, it's this long segmented bacteria. So it looks like kind of like a chain of cocci, but it's not, those aren't individual cells. That's all one cell.
Oh, yeah.
And it forms this really long chain, and it actually will kind of spiral into the mucosal layer of the gut. And a lot of work's been done in mice where they show that it stimulates a strong immune response and helps with developing gut immunity. Well, we found that birds that were performing better, if the flocks that had better performance, you would find this bacteria. It was a clear marker of the flock moving towards that state. We were really excited about that. The challenge with that one is it's really hard to culture. For a long time, it was non-culturable. Now there's some groups that are culturing it, but I think there's a lot of promise there, and I'm glad that people have continued to work with that bacteria. We kind of gave up. We tried culturing for a while and gave up on it, but it was exciting, and I think there is some promise there for sure.
Well, that's the great thing about science, is that somebody else can take some of your information and keep going. They've had experience culturing some of those other problematic bacteria.
Yeah.
So before we wrap up, is there anything that we missed? Any major points that I didn't ask you about that are really important for someone to understand the topic?
I think just one thing I'll bring up or just mention is I get a lot of questions about sample size and where should I sample, what should I sample, and there's actually a working group right now that will present at the Poultry Science Association meeting. I believe they've got a poster, but they're working really hard on trying to develop some standards for microbiome research in poultry. And I think that, and there's also a paper that's been written about in the Poultry Science Journal as well. But either way, if you have a chance, your audience has a chance to look at that, I think it's worthwhile because it provides some really good basic guidelines for if I'm going to do, I would say basic, but also realistic, right? Because a lot of people, if you ask them, what should I do? Well, you need 100 samples here and 100 samples there. And before you know, you're spending, $30,000. But they provide some really realistic and basic guidelines for doing things in a way that allows them to be repeated later, but also ways that you can do other things later and compare it to what you've done at the time. So I think it's a worthwhile thing to look at if somebody's thinking about the microbiome.
It's time for our famous three.
For over 40 years, Jeffo has been redefining animal nutrition with high-performance, non-medicated solutions. At the core is Jeffo Matrix technology, a disruptive approach that synchronizes nutrient release for precise delivery. Active in 65 countries, we develop cutting-edge formulas, essential oils, enzymes, and acidifiers to optimize health, performance, and reproduction. Jeffo is transforming animal feeding, making life easier for animals and people who care for them.
Yeah, that is great advice. Speaking of worthwhile resources, do you have any resources that somebody who's new at the microbiome or just wants to understand it better might look into if they're trying to increase their knowledge?
Yeah, if you're really just trying to get started and understand the microbiome, if you go to, and I don't have the link, but it's through PubMed, so NCBI, they have this entire resource on literature, materials, things that talk about the basics of the microbiome. I've sent a few students there in the past, and they said that they have some fantastic things that you can read about. So I think it's ncbi.nlm.nih.gov is the website. Yeah.
Awesome.
So if you go there, there's some really good materials.
Yeah, and I'm sure that's updating in real time too, so that's even better.
And from a poultry standpoint, there's a lot of pretty decent reviews about the microbiome out there. I won't name any specifically because I don't want to be biased, but
Yeah, I get it.
Really good overviews of the microbiome.
I get that. Totally get that. Have you been reading or listening to anything lately that's been really interesting?
I'm kind of a fan of the crime stuff, the podcast.
Yes.
So typically, like if I do a road trip, I'm listening to like the latest Dateline episodes or whatever, all those different podcasts that are out there now. And so many of them, I can't even remember them all, but
I know they just flip through to the next one when one ends.
Time passed quickly. So I do a lot of that.
Yes. Yeah. I'm on board. Sword and Scale. I love listening to them. What's your best advice for someone to be successful in the poultry industry?
For research, it's challenging when you're first getting started from the standpoint of trying to establish contacts, trying to become someone who's known, right? That's the goal, so that people because then they come to you for more research. And I think that, the best advice I ever got from my advisor was just to get out there and make your work seen. I mean, these are pretty common pieces of advice, but even like going to that meeting, whether it's Poultry Science or AAAP or any of the smaller meetings, not just going and sitting in the audience and presenting your work, but also taking the time to go up to that person that maybe you admire their work or you're curious about their work, and just say, hey, can I borrow you for a few minutes and ask you some questions? And you'd be really surprised at where that can lead. I mean, it might just lead nowhere and you'll just get answers to your questions, but it could also lead to collaborations and beyond that, just friendships that can last years, right? And so don't, I think that's the hardest thing for especially students to do, is to just put themselves out there and talk to people that they see as above them, but really nobody's above anybody. We're all just trying to, make things better for chickens and turkeys. So don't ever feel like you're inferior. Don't have that complex. What do they call it? The one where you always think you're not good enough. But
the imposter syndrome.
Imposter syndrome. That's it. Don't have the imposter syndrome. Just remember that everybody is just like you. So just go out there and talk to them.
I think that's great sage advice, especially coming up to the summer meeting schedules. A lot of meetings we'll be going to for us in academics and industry.
Yes.
Have a good chat with people who are there. They know something.
Yeah, it's not always just the scientists, right? You learn more from the people that are raising birds.
Oh, heck yeah.
Yeah, for sure.
Totally agree. Well, thank you so much for talking to me and the audience about the microbiome. I love hearing the recent updates. I know enough about it to be dangerous, and that's not. I like hearing the things that are new, upcoming, and advances. So thank you so much.
No, my pleasure. Thank you.
You're welcome. If you listen to this episode, share it with your fellow compadres who also are into poultry, so more people can learn about Dr. Johnson's work.